currently working as assistant professor in the Department of Botany at Al Muslim University. His current research interests are the selection of high-yielding mutant varieties in pulses using mutagens such as EMS, sodium aside, gamma rays individually or in combinations. Dr. Aamir has developed 11 high-yielding cowpea mutant lines. He has published more than 50 journal articles and 25 books chapters and two books. Considering all his achievements and outstanding contribution in the field of plant science, he has been conferred with various research fellowships. Dr. Amir has been awarded National Environmental Science Academy Young Scientist and the Bharat Vikas Award. He also has been recognized as Young Scientist of the Year 2018. Dr. Amir is among the editorial board members of many scientific journals such as Frontiers in Genetics, Scientific Reports, PLOS ONE, and PMC Plant Biology. Dr. Amir, thank you so much for joining us today and to deliver your outstanding research. And please start your presentation.
Thank you, Dr. Ahmed, for a nice introduction. I'm Dr. Aamir Raina, working as an assistant professor in the Department of Botany. I'm thankful to the Dr. Ahmed for providing me an opportunity to deliver and talk about my research, which is entitled as Role of Mutation Breeding in Crop Improvement and Impact of Coffee Mutant Varieties in Addressing the Food Security. So first of all, I will begin with the snapshot on induced mutagenesis. We have the two major pressures right now in the world. One is high population growth and another is extreme weather events. And among this, developing countries are the worst hit in meeting the food demands of their people, with millions unable to access adequate and nutritionally balanced food. Crop production must be increased by 70% to keep up with the food demands of the rapidly growing population, which is expected to rise to 9.6 billion by the end of 2015. This is as per the reports of Food and Agriculture Organization. So why we chose legumes or coffee? There are several reasons why we started with the coffee. Legumes are ideal crops to increase the agricultural food productivity and achieve sustainable developmental goals. Legumes are also climate resilient. Some of the legumes are heat tolerant, some are drought resistant, and others have other good traits which are important from farmers' perspective. Coffee, a warm season grain legume, is often categorized as neglected crop with immense scope for genetic improvement through proper breeding strategies. The annual average production of coffee is low compared to the other grain legumes, primarily due to narrow genetic variability. How we implemented it in coffee, we have some of the objectives, like we're interested in enhancing the genetic variability. We chose two coffee varieties which are named as Gomti VU89 and Pusa 578. We treated these varieties with different doses of gamma rays and sodium azide. In the literature, a conclusive evidence about the optimum dose of gamma rays and sodium azide is not available, and we have this kind of knowledge gap in this copy. A multi-year field experiment was conducted from April 2014 to October 2017. This is the flow chart in which I have tried to show the illustrations of induced mutagenesis and the selection of methodology followed during 2014 to 2017. So we began with the 4 doses of gamma rays like 100 GY. GY is the gray, which is the unit of radiations, 100 GY, 200 GY, 300 GY, and 400 GY. We also started with the sodium azide with combinations like 0.01%, 0.02%, 0.03%, and 0.04%. So we also use the combinations like we combined 100 GY gamma rays plus 0.01% assay, then 200 GY gamma rays plus 0.02% assay, so on and so forth. We also optimized the mutagen doses. A radio-sensitive test was conducted to optimize the mutagen doses. Separate seed sets of 300 dry seeds each were treated with gamma rays, 100 GY to 1000 GY, sodium 0.01% to 0.1% and their combinations. Details of the 2014 to 2017 field trials. The dose is beyond 400 GY in the gamma radiations and 0.04% in the sodium azide and 400 GY plus 0.04% assay caused more than 50% reduction in the seed germination. So we decided not to include all those doses beyond 400 and 0.04% and 400 plus 0.04% assay in our subsequent generations. So in the year first, which is 2014, during mid-April 2014, a total of 7,800 copy seeds were sown in 30 applications at the agriculture farm of Aligarh Muslim University, Aligarh, India. In the second year, 2015, 10 healthy seeds from each M1 plant. M indicates the mutagenized plant were sown to raise the M2 generation. Based on the quantitative statistics, 30 high-yielding mutant lines were isolated and advanced to subsequent M3 generations. In 2016, which was our third year, from the M2 generation onwards, we advanced only 30 high-yielding and non-segregating mutant lines to M3 and M4 generations. Sorry. The effective selection process led to the isolation of 11 high-yielding and non-segregating mutant lines. In the M4 generation, we raised only 11 high-yielding mutant lines and non-segregating mutant lines. We also conducted biochemical studies like nitrate reductase activity as per the protocol of Jaworski in 1971. We also checked the chlorophyll and carotenoid contents following the method of McKinney 1941. This slide shows the pictures of control plant of variety Gomti VU89. The B picture shows high yielding mutant line of variety Gomti VU89. This is the control plant of variety PUSA 578 and high yielding mutant variety of PUSA 578. Here you can see there's a visible difference in terms of the number of pods per plant. On the other side of this slide, we have tried to show which of the doses were effective in inducing these different mutant lines. Like we have Gomti VU89A, we have Gomti VU89B. So we named them as per the alphabets. Apart from the conventional markers, we also used molecular markers. In this, we use it SDS page analysis as per the protocol formulated by the Lamely 1970. We also used simple sequence repeat SSR and CAD box derived polymorphic, which is abbreviated as CBDP marker analysis. The leaf DNA was extracted using CTAB, cetyl trimethyl ammonium bromide method. In our this present study, 82 gene SSR and 25 CBDP primers were screened, and finally 9 primers led to the generation of polymorphic bands. What we observed and recorded, the different doses of gamma rays and sodium azide induced a substantial variations in the morphological traits of copy. Quantitative trait analysis revealed maximum increase in the seed yield from M2 to M3 generations. Seeds per pod and seed weight positively correlated with a major impact on yield. As far as the statistics is concerned, one way ANOVA revealed a significant increase in the mean values of almost all quantitative traits except seeds per pod in both the varieties and seed weight in variety PUSA 578. This is attributed to the random nature of the mutations. It's a hit and trial method in which we can get both favorable results as well as unfavorable results. This slide shows the pictures. In this, we have the A, which represents the experimental field at the seedling stage. Then B represents the experimental field at this harvesting stage. C represents tall mutant. D is the semi-dwarf mutant. E is the bushy mutant. F represents the variations in leaf size. G to H represents the variations in the flower color. I represents variations in the pod length. J represents variations in the pod number. K and L variations in the seed size, and M and N indicates variations in the seed coat. What were our key observations? In our study, lower and medium doses of mutagens were more effective in inducing in increasing the mean values of important quantitative traits such as spots per plant, seeds per plant, and seed weight and seed yield. A substantial increase in pods per plant was recorded in the advanced generations. Interrelationships among yield and yield component traits. In mutation breeding programs, correlation analysis is very important as it helps us to visualize the relationships between yield and yield attributing traits. In our study, plant yield correlated positively with all the studied traits except days to flowering, which is also good for us because during the this flowering stage we have a lot of this wind which could be detrimental for the plants. So these plant yield showed a negative correlation with the based flowering. The negative correlation between yield and flowering time is also an important trait of interest in the warm season legumes like coffee. This slide shows correlation analysis between yield and yield attributing traits. GOM TVU 89 and B represents variety PUSA 578. PY indicates plant yield, triple P indicates pods per plant, BPP branches per plant, and SPP seeds per pod. SW is seed weight and PL is pod length. HI is harvest index. Significant differences are indicated as P greater than 0.001 or P less than 0.01 and P less than 0.05. So here we have there are some numbers which are associated with different traits like we have plant height, then days of flowering, then days to maturity, pods per plant, branches per plant, and seeds per pod, seed weight, pod length, plant yield, and harvest index. We also try to analyze the body biofortification in the high-yielding coffee mirror lines. In our study, a concurrent increase in the yield and nutrient density in M4 high-yielding mirror lines indicates a broad possibility of developing biofortified coffee, providing an added benefit in the current scenario of food insecurity and widespread malnutrition. Biofortified coffee varieties could keep up with the nutritional demands of millions of undernourished people in countries. We also implemented path analysis to help us in visualizing the direct and indirect impact of component traits on the yield. It provides a precise relationship between yield and yield components. The path analysis revealed that seed weight and seeds per pod are one of the primary yield components due to its greatest direct and indirect impact on the plant yield. This slide shows the path analysis in the variety home TVU 89 and variety PUSA 578, showing an interrelationship between plant yield and yield attributing traits. On the other side, we have dendrogram clustering based on quantitative traits in both the varieties. We also studied multivariate analysis. In our study, hierarchical cluster analysis divided mutagenized and controlled populations into separate clusters. Genetically diverse populations were grouped within the different clusters. Therefore, mutants selected from different clusters could be advanced to subsequent generations. We also checked principal component analysis, which showed significant positive correlation between plant yield and pods per plant, seeds per pod, and branches per plant. Days to flowering correlated negatively with the yield and yield attributing traits, suggesting that earliness is a desirable trait in breeding programs. This is the pictorial representation of the PCA. We also studied or analyzed the nitrate reductase activity and pigment contents. Activity of nitrate reductase enzyme and the contents of chlorophyll and carotenoid proteins and mineral contents were significantly high in the selected mutant lines compared to their control population. This slide shows mean values of spots per plant yield, nitrate reductase activity, chlorophyll, carotenoid, proteins, iron, copper, zinc, in 11 M4 high-yielding mutant lines and control plants. We also studied molecular characterization using SDS page markers. SDS page profile of seed proteins generated 54 and 28 polymorphic bands, and the total polymorphism of 62.06 and 41.17% in Gomti VU89 and PUSA 578 respectively. SSR primers amplified a total of 16 and 24 polymorphic bands with an average polymorphism of 20.69 and 50.74% in Gomti VU89 and PUSA 578 respectively. CBDP markers generated 175 bands, with 77 bands being polymorphic in Gomti VU89 and 121 bands with 59 bands being polymorphic in PUSA 578. This slide shows the SDS page profile showing the bands of proteins and control proteins of control and M4 hiding mutants of copy varieties. So this is the SDS profile of WOM-TV89, and this is SDS profile of PUSA 578. Here you can see a lot of this polymorphism, that these uneven bands are present in both the varieties. And on the other side, we have dendrogram analysis of control and 7M4 hiding mutant lines based on SDS page profile protein samples of copy variety GOM-TV89. This slide shows the SSR molecular markers and detection of DNA polymorphism. Here you can see it's very visible. A lot of polymorphism is there in the POSA 578, and this lane represents the control, and this represents the mutant first, which is GOM-TV89A. This is second, GOM-TV89B, and so on. And this is the marker. This represents the letter or the marker. So here we have a lot of polymorphism in the PUSA 578. Here we have polymorphic bands in the GOM-TV89. Here again we have polymorphic bands. Again we have polymorphic bands. So this is this VU1. It represents the this SSR marker. VU2 is second SSR marker, and VU10 is third SSR marker. We used in total 82 genic SSR markers. And this slide shows the CBDB molecular markers and detection of DNA polymorphism. Again, here we can see the DNA polymorphic bands in both the varieties. Here it's more visible in case of CBDB 13. CBDB 18 also is also good. So this was the study about molecular characterization of the copy mutant lines. In conclusion and future directions, the concurrent research led to the development of high-yielding copy mutant lines, and the seeds of mutant lines are stored in the department seed bank for further farmers' field-based characterization and dissemination to the copy breeders. The data on quantitative traits indicates that mutagens were effective in inducing substantial interpopulation genetic divergence. The third marker analysis revealed expression of new polymorphic protein bands among the high-yielding mutant lines. In the M4 generation, high-yielding mutant lines showed improved genetic gain and increased nutrient density, and can serve as parents in the crossbreeding programs to broaden the genetic base of the copies. I'm thankful to the audience for listening to my lecture, and thankful to the Dr. Ahmed for providing me this opportunity to talk about my research.
Thank you, Dr. Amr, for this really interesting lecture and useful information. And now we open discussion. So anyone of the audience would like to ask questions, please raise your hand, and I will allow your mic. Any questions for the attendees? Okay, I may start, Dr. Aamir, for the questions. I would like to ask about which mutagens is more effective, AMS or gamma rays, or and you didn't give us like concentrations that we can use. You mentioned that low concentrations may be very effective to get like variations among the plants. So could you please expand this information to us?
Yeah, it's a good question. As per the reports of MVD, which is Mutant Variety Database, it is maintained by the IAE, Austria, in collaboration with the FAO. So this database shows that gamma rays are more potent in inducing the mutations, and till now more than 1600 varieties out of almost 3,500 varieties have been induced by the use of gamma rays. And as far as the second part of the question is concerned, which of the dose is going to be good for us? So it depends on the plants. So some of the plants are highly sensitive to the mutagen doses. So we usually use lesser doses like 25 GY, 50 GY, and 75 GY doses of the gamma rays. Some are like pulses; they are not as sensitive as oilseed crops. So we can start from the lower dose as 100 GY, then we can go up to 500, 600. That doses have also been shown to be good for the mutation breeding. So it depends on the plant that we are working on.
Okay, and what about faba beans? Because I'm interested in faba bean crop. Did you work on faba bean before for you tried for mutagens in this crop or only
Yeah, yeah.
cowpea and
Yeah, we our research group is basically focusing on the legumes. We have worked on several plants like chickpea, faba bean, and this lentil, cowpea, some black bean, or these some species of the vigna genus. So we basically focus on the legumes, but over the past few years, we have tried to shift our focus to the medicinal plants. In medicinal plants, we completed the experimentation or the projects on the Nigella plant, and we also try to analyze the effects of different mutagens on fenugreek, and we also studied effects of mutations on Trichospermum emi. So right now we are trying to study the effects of gamma rays and sodium azide on this buckwheat. So it's I can say that we have completed the mutation breeding in the pulses, and we have succeeded in producing some good mutants which are in under trials. Right now we are producing the M5 generation of the copy, and if we get a desirable results, we'll then shift towards the multi-location trials, and finally we may succeed in releasing the this highly muted variety of copy.
okay. And
Yeah,
just another question. Did you check the quality, grain quality, for this mutant variety? I mean, compared to the original one.
yeah.
Um,
Yeah,
yeah.
there are a lot of traits like there are quantitative traits and qualitative traits. This grain quality comes under qualitative traits, but in our this lab we focus on quantitative traits.
Ah,
So
okay.
like we are interested in all the quantitative traits like pods per plant, number of seeds per pod, seed weight, and total yield, but we have not studied the grain quality, the effects.
But
But
me,
it seems interesting to
yeah,
analyze the effect
yeah.
of mutations on the grain quality as well.
Yeah, because in my point of view, is just you get a mutant in like in some genes. So yeah, maybe you improved some traits, but maybe this mutant affected other important traits like green quality. So maybe I got good plant, but maybe the greens uh, don't taste enough like for consumers or something like that. Okay, thank you for
Yeah,
answering the questions.
thank you,
So,
Dr. Ahmed, and
yeah,
yeah,
thank you.
yeah,
Do
thank you for providing me this opportunity.
thank you so much. You can write your questions if you can't use your mic. You can to write your question in the chat room, and if any one of the attendees has a questions regarding to the lecture,
Yeah, yeah, and I'll just check.
yeah, I think, yeah, I think no, we don't.
It seems no questions. Yeah,
Yeah, okay. So, come to the end of the lecture. So, thank you, Dr. Amir.
you are
Um,
welcome.
yeah, for this lecture, and I hope that we can collaborate in future for this topic.
Yeah, inshallah, we will
Inshallah.
definitely collaborate. Inshallah,
Okay.
yeah.
Thank you, Dr. Amr.
Thank you,
Uh,
Dr. Ahmed.
Thank
Thank
you.
you.
Thanks for the attendees, and we will send the link for instruction how to get the certificate of attendance. Thank you, Dr. Amr. Thank you.
Thank you. Thank you.
Have a nice day.